Welcome, everyone, and thank you for joining us today. We're excited to have you here for our webinar, bringing together multi-omics and real-world data to accelerate insight delivery for biomarker discovery and drug development. I'm Felicia Sabatino. I am a Senior Project Manager at Rancho BioSciences, and I'll be your moderator for today. Before we get started, I would like to note that today's session is being recorded. We'll be sharing the recording with all registrants after the webinar, so you'll have a chance to revisit the content and or share it with colleagues who are unable to attend. We have time scheduled at the end of the call today for a Q&A session. We would absolutely love to hear your questions on today's presentation. However, we ask that you use the Q&A panel to add your questions, not the chat. At the end of the webinar, we'll do our best to go through and answer all of those questions. And then, all right. Let's take a quick look at what we'll be covering today during the presentation. After I share a little bit on the collaboration between Rancho and Sapient, we'll start by meeting our speakers. Next, we'll dive into the challenges and success factors associated with the lakehouse. After that, we'll take a look behind the scenes at the methodology that went into the build. Then we'll look into some of the practical use cases that this opened for us. And finally, we'll save some time at the end, as I mentioned, for your questions. So please don't hesitate to hit that Q&A panel throughout the session. Before I introduce today's speakers, I'd like to take a moment to introduce the collaboration between Rancho BioSciences and Sapient that brought us here today. Those of you who are familiar with Sapient know their impressive in-house multi-omics database already holds metabolomic, lipidomic, and proteomic data from tens of thousands of human biosamples, plus millions of paired phenotypic data points. This wealth of data allows Sapient to help clients validate biomarkers, identify drug targets, and gain vital biologic insights. Rancho BioSciences' collaboration with Sapient builds on that strong foundation, leveraging our deep expertise in data curation and knowledge management to support the expansion of Sapient's database infrastructure into a next-generation insights engine. This means we'll see even faster ingestion and homogenization of new multi-omics and real-world data. This partnership is designed to deliver even broader, deeper, and more multidimensional insights to inform discovery and drug development. It truly is a powerful pairing, bringing together Sapient's cutting-edge multi-omics data generation and AI-driven insights with Rancho BioSciences' data science expertise. We're all very excited about what this means for accelerating drug discovery and ultimately improving patient outcomes. Now, let's get started with meeting our presenters. All right, today's presenters, I'm pleased to introduce you to Tao Long. Tao Long is the co-founder and head of data science at Sapient. Dr. Long is a senior bioinformatician and computational biologist with over a decade of experience in handling and analysis of multi-omics human data sets, including genomics, proteomics, and metabolomics. Prior to co-founding Sapient, she was the head of bioinformatics at UCSD. While at Sapient, she leads all data science initiatives and is responsible for maintaining and expanding Sapient's proprietary human biology data assets. Next, we have Jamie Cho. He is the head of software at Sapient. Jamie is a senior software developer with over 25 years of deep software engineering experience in the life sciences industry. He was formerly head of software at Ginkgo Bioworks and was previously director of applied research at Bruker. While at Sapient, Jamie leads the development of software solutions that optimize operational efficiencies and enable maximal extraction of insights from complex at-scale biological data sets. Joining us from Rancho, we have Elena Fedorovich. Elena is a senior principal scientist and head of the data harmonization and knowledge mining group at Rancho BioSciences. Dr. Fedorovich has over 30 years of experience in life sciences industry in research and development. Before joining Rancho, she was an assistant professor at the biochemistry department and molecular biology departments at Medical University of South Carolina. She has a solid track record of peer-reviewed publications. Over the past decade at Rancho, Elena has been deeply engaged in biomedical data curation and has successfully led numerous complex data harmonization projects. Her work focuses on applying FAIR data principles and draws upon her subject matter expertise across a range of clinical and real-world domains. Finally, joining us from Rancho is Marjolaine Spronk. Dr. Spronk is a principal scientist and team lead in the Data Empowerment and Innovation Group at Rancho BioSciences, with more than a decade of experience in processing and analysis of life science data. Prior to joining Rancho, she completed postdoctoral fellowships in imaging and advanced training in data science. At Rancho BioSciences, Marjolaine combines data science methods with life science expertise to provide robust data solutions. I'll now hand it over to Tao, who's going to get us started with the data lakehouse overview. Tao?
Hello, everyone. Next slide. I want to start by briefly introducing Sapient. We are a discovery lab headquartered in San Diego, and we specialize in multi-omics data generation and insight delivery for drug development. We innovated mass spectrometry technologies to profile thousands of proteins and metabolites across thousands of samples at a time. And these technologies are unprecedented in both the scale in terms of number of molecules measured and the throughput. Beyond multi-omics data generation, we also provide deep biocomputational expertise to integrate and make sense of these large-scale datasets, allowing us to uncover novel biological insights. Sapient's omic work is particularly centered on the discovery of dynamic biomarkers that can read out changes in biological process, disease, and drug response. We generate this multi-omics data to build upon the significant efforts made in genomics field over the past few decades. While genomics has had a transformative impact on drug development, it has become clear that genetics alone tells only part of the story. It has long been appreciated that dynamic factors such as proteins and metabolites can have far greater influence on overall health outcomes. And so our goal is to layer on these additional readouts through proteomics and metabolomics analysis to provide a more complete view of human biology and how disease manifests, progresses, and responds to treatment over time. Proteins, metabolites, and lipids reflect both endogenous and exogenous influences and elucidate molecular mechanism underlying the diseases, can change dynamically with disease progression and treatment, and thus also provide direct targets for therapeutic intervention. Since the beginning of Sapient, our focus has been not only on generating this data, but also leveraging this for delivering actionable insight. So we established what we called our human biology database, which is an aggregation of these large-scale multi-omics datasets generated in-house and the accompanying real-world electronic health record data. You can imagine the complexity and the scale of this data asset, as we are able to generate over 12,000 proteins in human tissue, 5,000 proteins in human plasma, over 15,000 metabolites and lipids, and over 350 cytokines and chemokines in human plasma across thousands of samples at a time. And these large omics data are linked to real-world data, including millions of data points for diagnosis, medications, lab measures, and demographics, etc. And so our human biology database comprises of large-scale multi-omics datasets and the millions of phenotypic and clinical data points from tens of thousands of samples. As we continue to generate and aggregate these massive datasets, we had a growing need to expand our data infrastructure into a next generation solution that would allow us to, one, accelerate the access of this multi-omics data and real-world data, as these datasets are scattered and of different formats. And two, to homogenize these datasets to increase discovery power and to allow us to compare apples to apples. We want a database infrastructure that allow us to perform analysis and deliver insights with greater speed, versatility, and depth. And ultimately, we want to ensure the high quality of these datasets, as we truly believe the quality of data going in dictates the quality of insights coming out. Now, we will have Jamie talk about how we pursued the effort to develop our next generation data lakehouse.
Thank you. When we were considering the next generation data lakehouse, there were three considerations that we focused on for real-world data. One is we wanted to make sure that the data is complete. We wanted to make sure all of the data sets are in the same format and in the same place so that scientists don't have to worry about, is my data on an Excel file in S3 or a CSV somewhere on a file share? We wanted to simplify the process so all of the data is in a single place and in the same format to make it as easy as possible to access. Two, we wanted to make sure that the data is consistent and harmonized to the same standards. So this is making sure everything is apples to apples versus apples to pears. And in particular, there are times where units can get mixed and matched very easily, such as pounds and kilograms. And we want scientists to focus on the data analysis without having to worry about performing these conversions. Three is we wanted to make sure that we handled as much of the data context as possible so that the scientists don't have to worry about that. And the data context is making sure that we have the most pristine transformations in place, making sure that we're handling all of the security concerns, and also making sure that all privacy concerns are considered as well. And really, we want to focus on these three critical success factors so that the scientists can ultimately focus on the data analysis without having to worry about these different confounding factors. In addition to real-world data, there are multi-omics data considerations as well. And in particular, we wanted to make sure that we built a system in which we could also build interoperable frameworks so that we can apply the same set of algorithms across different kinds of data. So simple examples are data normalization and imputation. We want to make sure that scientists can apply the same set of algorithms to the different sets of data so that, again, that they can focus on the work at hand versus having to learn the nuances of this framework or that framework. Two is we wanted to make sure that our system that we're building is scalable. Right now, we integrate many different kinds of technologies, and we want to make sure that we can continue building and integrating different sets of data as they become available. So going from, for example, metabolomics to proteomics to genomics, we want to make sure that we have everything supported in this data lakehouse. Three is we wanted to make sure it enabled interpretation. And ultimately, what we want to achieve is connecting different kinds of data together and linking them up and being able to analyze them using the real-world data as the connection point. And ultimately, what we're trying to do is enable meaningful biological interpretation through this robust set of analytic frameworks. And as we were considering these things, we decided on partnering. And the reason why is there are lots of tools out there and lots of specialized interests that we want to tap into. And in particular, there are advanced AI tools that are available for data harmonization, as well as a lot of domain expertise. And we looked at both. So looking at the advanced AI tools, what's great about those is that they enable curation for huge sets of data, hundreds of millions of records that can be done in seconds. And that's very powerful. But the problem is that they lack a lot of context that people have. And as a result, they have a tendency of mixing and matching units such as kilograms and pounds. And that yields results that are subpar and not up to the standards that we're looking for. And so the next consideration is let's double down on domain expertise. Domain expertise will ensure that we have high quality data and high quality results. The problem is that we have hundreds of millions of records, and curating each of those by hand is very onerous, and it'll be ultimately too manual and too slow. And so when we were looking for partners, we wanted to make sure that we could partner with someone that had a mastery of both advanced AI tools, so enough mastery so that they have their own sets of tools, as well as a lot of domain expertise so that they can apply those tools. And with Rancho, we found a perfect partner that has both the deep domain expertise, but also the advanced set of tools so that they can do pristine harmonization work with in a very cost-effective manner. And with that, I want to hand it over to Marjolaine, who will talk about the details of their process.
All right. Thank you for that, Jamie. So we'll be moving on to the methodology. Marjolaine is going to give us a behind the scenes insights to the building that went into this database. Marjolaine?
Yeah, thank you, Jamie and Felicia. So here you can see our end-to-end integration framework for this collaboration, where we bring together the real-world data and the complex omics data from a variety of sources, such as demographics, medications, outcomes, and lab data. The data from these sources differs in format, in structure, in scale, from clinical records and lab results, for example, to high-throughput omics data. To enable interoperability between these types of data and between studies, we aligned the structured sample and patient metadata to a common data model, which you see in the middle here. By doing this, we can give consistent meaning to key elements in the data, even though they come from very different sources. And this makes it possible to reliably connect clinical events with specimen data. We can ingest both structured and unstructured data into a data lakehouse. So that means we can store the raw data, but also structured tables all in one place. And that gives people access to the data as needed for their specific use cases, for their analysis and visualizations. This creates a governed environment that can support exploratory research, cohort building, etc., without having to work with silo data, for which users would need to create separate scripts to, for example, join the different types of data from different sources. So it makes it much easier to work with the data, and it accelerates insight delivery by providing quick access to high quality integrated data. Now that we've outlined this overall framework, let's take a closer look at what the common data model looks like that made this possible. So our next step was to design a conceptual model that is aligned with the OMOP model. For those not familiar with OMOP, this is a standardized data model that is designed to harmonize healthcare data from different sources. So it can be analyzed in a consistent and reproducible way. And it is used by hundreds of institutions and organizations. It's become one of the most widely adopted standards for real-world data. We wanted to leverage this standard, but only in places where it really makes sense, where it fits. So mostly for the clinical data. To accommodate the omics data, on the other hand, we customized the model instead. Another reason for aligning to OMOP but not fully adopting OMOP is that OMOP can get quite complex, and that complexity may not be needed everywhere. We wanted to keep it focused only on the relevant sections. That makes it easier to implement and maintain the database, but it's still compatible with the OMOP standards. So that's what you see here. Our conceptual model is person-centered like OMOP, but also accommodates the omics data that you see in the orange parts here. By organizing the data as we did here, we can manage complex big data, reducing data silos, and make the data more easily accessible. With many data standards out there to choose from, you may be wondering why OMOP. So let's take a look at that. There are several reasons why we chose to align with an OMOP-like common data model. So interoperability and standardization is one. By organizing the data around a shared schema, we can ensure that the data from different systems, whether electronic health records, registries, or research files, can be integrated and understood in a consistent way. In other words, it enables that apples-to-apples comparison between different systems. Second, OMOP provides standardized vocabularies and mappings across clinical domains. These include, for example, SNOMED, BLOINC, RxNorm, and others, which allows us to translate raw data terms into globally recognized terms. Each concept in OMOP is assigned a unique concept idea with predefined mapping across vocabularies. So ICD-10 to SNOMED, for example. Without OMOP, a diagnosis like major depressive disorder could be represented in many ways, but with OMOP, they all resolve to a single concept idea. Also, the model is designed to support scalability for population-level analytics. OMOP's standardized structure is designed to scale, making it just as effective for analyzing small patient groups as for conducting very large-scale population studies, all while still enabling efficient querying, consistent cohort definitions, and reproducible workflows. Many databases can support OMOP structured data. And last, aligning with OMOP helps us and others ensure that your data is structured in a way that meets the expectations of both regulators and the broader research community. These standards are more and more recognized by, for example, the FDA and enable easier participation in collaborative research networks, many of which already use OMOP standards. So overall, using OMOP ensures that your data is fair and scalable. But to get there, the data must first undergo transformation, of course. So to help with this, we first conducted a data review of the source data. This included the data completeness and coverage assessment, where we evaluated how consistently each variable was populated and across how many records, helping us really understand both the gaps and the strengths in the data. Based on this review of basically all the available data in the different buckets, we selected the most relevant variables for integration, prioritizing those with high completeness, consistency across files, and of course, analytical value for the user. We then used this refined variable to define a variable set to define the attributes and structure needed in our common data model, making sure that the model would reflect the actual data available while staying aligned with standard conventions. So this upfront data review work is really critical in guiding the modeling and the ETL design. It made sure that the final model was both practical and fit for purpose, and the data was ready for further harmonization and transformation. So after defining requirements for the common data model, we needed to further expand and create a logical data model. So we translated our findings into a logical data model that could actually guide the database design and the ETL process. So a logical model defines specific tables, fields, data types, and relationships, giving structure to how the data will be stored and linked. As mentioned before, we aligned to OMOP, OMOP's core tables and variables to represent real-world data elements like patients, visits, clinical observations. But because OMOP does not fully support omics data, we extended the model with custom tables and attributes to capture specimen data, assay results, and other domain-specific information. And that hybrid approach really allowed us to benefit from OMOP standardization and vocabulary infrastructure while still capturing the granularity and complexity needed for the multi-omics parts. This model served as the blueprint for our ETL design, ensuring that both standardized and custom data elements could be ingested consistently and linked across domains. It also made downstream querying and analysis more reliable, especially when combining clinical and omics data. So by conducting the data review and creating the logical model, we can better prepare for data harmonization as it helps us avoid surprises later on. After finalizing the data model, we moved into the harmonization of the actual data. So I will now hand it over to Elena, Rancho's expert curator, to walk us through that harmonization and curation process.
Thank you, Marjolaine. So using the created data model, we began our data curation process. 7 key data model entities were selected for data harmonization and curation based on relevance and impact for analysis. This included person with demographic information, condition for diagnosis, drug exposure for medications and prescriptions, procedure for medical interventions and procedural treatments, measurement for lab results and vital signs, observations with additional demographics, non-numeric lab values, etc., and lastly, specimen that links clinical and omics data. The emphasis was placed on 16 attributes spanning for 7 core entities. To transform data into a consistent data set, unique terms were extracted from each data source. Variability in data formats and definitions, even within the same source, made us to develop custom rules for each data type, cleaning, curation, standardization, and alignment to OMOP vocabularies. The combination of automated and manual curation aimed to establish a strong analytical foundation focusing on the most relevant and high-impact data. This is a high-level model here, but dealing with real-world data is much more complex, and there are many ways to iterate various data categories, including conditions and diseases. For conditions and procedures, raw data codes were mapped using OMOP code-to-code mappings, which were manually reviewed and adjusted using raw descriptions. Multiple descriptions of the same disease originally assigned to one or several ICD-10 and or ICD-9 codes were standardized to one standard term. There were tens of thousands raw descriptions that were standardized. For example, the 32 original real-world terms could be harmonized to one standard term, cyanosis of liver, as you can see on the screen. This helped to further categorize conditions by disease class. Diagnoses were further grouped in classes to make cohort stratification more user-friendly. As a result, a significant reduction of redundancy and variability in terminology was achieved through curation efforts by our expert curators. Curation was expedited by using automation, as well as we'll see next. Source medication and prescription data used for drug exposure domain provided several variables from which 2 were selected for curation. This included drug name with inconsistent unclean drug descriptions, including brand names, dose, form, with misspellings, abbreviations, spaces between words, and so on, and corresponding generic drug name. Raw generic drug names were first automatically mapped to OMOP using Rancho Terminology Management Solution, or TMS tool. A list of more than 1,000 raw terms uploaded in the TMS as CSV file and was further returned as an output table with mapped terms. The automated alignment was followed by manual review and adjustment, including correcting inconsistencies found between drug descriptions and generic names, helping general data quality assessment as well. Using TMS saved time and increased accuracy, leading to less artifacts, outliers, and greater insights. Here's a quick overview of what makes TMS so effective. Rancho TMS is an API suite that leverages our more than a decade expertise in terminology management, data curation, data engineering, and artificial intelligence to accurately map any term or entity in the life science space to about 40 preloaded ontologies using phonetic and semantic algorithms. The tool has a simple user-friendly interface and easy workflow for term mapping to an ontology or ontologies of choice. The endpoint returns map terms along with query and synonyms. The results are ranked by similarity score for simple programmatic prioritization. TMS can be seamlessly integrated into any data harmonization and verification pipelines and curation workflows. Let's look at accuracy. We have a benchmark we'd like to share. Using TMS, about 2,000 source drug generic names were mapped to standard OMOP drug ingredient class, usually based on Rx norm vocabulary. This reduced manual curation time by 50% and resulted in an output of 65% high-quality matches defined by similarity score. These are a few examples of drug names mapped to AMOP drug ingredients using TMS. The tool allows selection of the most relevant ingredient by choosing the highest scoring term. Unique generic drug names were mapped to their corresponding ingredients using a mapping algorithm, which provides similarity score outputs ranging from 0 to 1, with 1 indicating an exact match or a recognized synonym in the AMOP terminology system. Manual review of the automated mapping confirmed the accuracy of the ingredients mapped to the corresponding source generic drug names. This also helped with the QC and adjustments of ingredients for 45,000 unique original drug names. The histogram on the right represents the frequency of similarity scores on the left axis in gray color across the real-world data set. And the kernel density estimation curve in orange provides a smooth probability distribution of the similarity scores. A clear peak at similarity score 1 indicates accurate mappings where the tool identified exact matches. Distribution of these similarity scores across generic drug names from TMS mapping confirms the accuracy of automated mapping. Once accuracy confirmed, map terms were organized into mapping tables, which were complemented by the development of transformation rules to pre-process and validate source data prior to ingestion into the data lakehouse. For instance, combination of automated set of rules was especially necessary for person measurement and observation entities. Information coming from multiple variables with defined terms or yes-no indicators or free text details was sorted into a single attribute in one or two entities like ethnicity, which became part of person demographics and observation. Vitals and general test names combined with specimen were mapped to OMOP terms, such as weight became body weight. Units were defined for each measurement and standardized to AMOP units where possible. Python-based set of rules was used to clean, transform, and integrate data containing numerous artifacts and unknowns. As general approach for numeric measurements, any outliers or ambiguous results or units were omitted from harmonization. Body weight here is an example. Raw data weight has different units or no units at all. Plotting weight frequently distribution helps to evaluate measurements without units and define transformation approach or rule. If a data point has no designation as a kilogram or pounds, and it can be either by value, it is removed from curated measurements. If there is an artifact of an outlier data point, it is also removed. Pounds are converted to kilograms where possible. However, importantly, to mention that all raw values are still loaded in the database and are available to scientists who want to access all data points. Once the data is fully prepared, it can be loaded into the data lakehouse. Now I'm going to hand this over to Jamie to walk us through the data ingestion process. Please go ahead, Jamie.
Thank you. So in building out the data lakehouse, there are different milestones and steps along the way to consider. There's data generation, data curation, data accessibility, and at-scale analytics. And each step along the way, these enable different outcomes, such as generating actual data to begin with, harmonizing the data, building out methods to make the data more accessible, and enabling that kind of at-scale analytics so that greater insights can be gathered without a lot of effort. And while we built a lot of proprietary tools and technology each step along the way, we're also building on well-established tools from different companies, including Bruker, Epic, Databricks, Tableau, and of course, all of the great harmonization work from Rancho. And ultimately, this enabled us, with our collaboration with Rancho on the next slide, to launch our next generation database. And This is ultimately allowing us to offer it as an expanded and enhanced service line for our pharma and biotech customers. We've branded this joint effort as Sapient's Dynamic Insights Engine, and you can see it holds multi-omics and real-world data from more than 56,000 biosamples collected longitudinally from a diverse population of more than 12,000 individuals, representing more than 60 diseases. We can now easily visualize this data for diverse analyses and support different areas of the drug development pipeline, from early target identification to biomarker characterization and the development of companion diagnostics. So as you can see, we represent a large number of diseases within Dynamic and a large number of samples, and those numbers are continuously growing as we add more data. Crucially, though, it was the curation and harmonization efforts that Rancho was able to help us attribute a greater number of samples to certain disease states. How? We may get harmonized data to, ICD-9 or ICD-10 or some custom framework, and that makes it really hard for us to know, oh, this sample actually represents both cancer and neurodegenerative, because different terms are used or because sometimes information is in different fields. And ultimately, by harmonizing all of this information to OMOP, we are able to more easily find samples that pertain to these different diseases. And this is where that prior focus on completeness, consistency, and context really does matter. We want to make sure that we have a complete set of information so that we can analyze the data in an unbiased way. And with that, Tao is going to walk us through how Dynamic helps us glean more insights.
Now with Dynamic, we can much more easily query and filter data and perform a variety of analysis and visualizations. For example, we can readily build a cohort of individuals with a specific condition of interest on certain medications and with longitudinal sampling over a certain period of time, and then evaluate the similarities and the differences in their patient journeys, including how their biomarker levels change with disease progression and the treatment interventions. We can also directly identify drug targets with proteomics data, and then we see a huge potential in oncology. Additionally, with this organized omics data and the clinical data, we can evaluate biomarkers and their associations with clinical phenotypes, genotypes, and environmental exposures. To dive into the biomarker insights a bit further, the key is that we can look at the biomarkers in the context of an individual's real-world experience. Having the clinical data matched with the molecular data over multiple time points allows us to understand the biomarker's stability and the dynamics across time, and treatment and exposure. We can get these views at population scale, and providing us enough statistical power for unbiased discovery of biomarkers that can denote responders versus non-responders, or those that may be likely to have any adverse events to particular treatments. We can also see biomarker patterns of early disease to better predict the progression and enable earlier intervention. So I want to show one real-world example in which we did just that, to identify an early diagnostic biomarker of metabolic dysfunction associated steatohepatitis, or MASH. With harmonized clinical data and matched metabolomics data in more than 10,000 samples from a diverse population, we identified an initial set of biomarkers associated with MASH compared to the healthy controls. Next, we prioritized the one biomarker that represents a few biomarkers within this set that share similar chemical structure that may reflect the underlying biological pathway. And then we look at this particular biomarker in MASH patients. And we found that this biomarker is elevated with the increased biopsy-proven disease severity. Interestingly, this biomarker is also conserved in a preclinical study where the level increased in the group of mice over the course of developing MASH, but not in the group of mice developing metabolic dysfunction-associated fatty liver disease, or MFOD. As shown in the Kaplan-Meier cumulative event plot on the left, individuals with high level of this MASH biomarker are at a higher risk of developing MASH. And elevation of this biomarker can be detected as early as a decade prior to the formal diagnosis of MASH. And on the right, it shows that this biomarker decreased within just 2 weeks post-bariatric surgery in individuals whose liver function was improved through the surgery, indicating that this biomarker can also be used to monitor response to therapeutic intervention. In summary, this case study showcases how we can leverage the dynamic database to rapidly identify robust, dynamic, and actionable biomarkers that can directly impact drug development, intervention, and treatment strategies. This is just one example of the discoveries we can make. Thanks to the analysis-ready data, Rancho has expertly integrated and curated. It's been an exciting collaboration and opens substantially more opportunities to collaborate with our pharma and biotech clients to generate insights that can drive their programs forward. Thank you, everyone, for your attention.
Thank you so much, Tao. Thank you to all of our presenters. That it concludes our presentation for the webinar on how aligning data to common data model helps create strong data foundation, enabling large interoperability data sets that can be consistently analyzed, leveraging that data-driven discovery and greater insights. We'll now open up the Q&A session. While we're waiting for questions to roll in, I'd love to tell you a little bit more about Rancho BioSciences. At Rancho, we are on a mission to save lives through data. We accomplish this by bridging science and technology with our subject matter experts. Rancho started as a data curation company, but has since grown to support our clients in many other ways, offering bioinformatics analysis, data harmonization, ontology mapping, data modeling, and custom tool development. Additionally, we aim to guide our clients towards the best strategy to meet their needs. At the end of the day, we're here to help our clients make faster, smarter, and more reliable decisions that ultimately advance science, benefit patients, and save lives. We'd love to discuss your use cases, so please feel free to reach out and schedule a free consultation. Alrighty, I see we have a few questions that rolled in, so let's get started. First one here, beyond OMOP alignment, what were the most challenging real-world data quality issues encountered during harmonization, and how were they resolved?
I think I can take that one. That's a great question. And yes, real-world data is challenging. So first of all, real-world data is really big, right? There's a lot of data. So processing-wise, that can really be a challenge. That's why we did a data review, so to make a clear distinction between what's relevant and what's not, and to make it all manageable. Another big hurdle, I think, with real-world data is missing or incomplete data. For example, dates may not always be available, maybe missing in some cases. Sometimes we can cross-reference other tables and get that information, but often it's just not there, and then we would not use it if their model requires that field to be filled. And then different sites may use like different names for the same type of information. Or we also see a lot of differences in granularity from different fields. That describe the same type of data, for example, diagnosis. That's something where you just have to carefully assess like what the needs are based on different use cases and align your harmonization to those requirements, I would say.
And I think I wanted to add here that even within a single data source, the inherent variability of real-world data poses significant challenges for standardization and often makes it difficult to preserve critical information. So you have to pay very specific attention to various types of data there. For instance, laboratory tests and vital signs in real-world data sets frequently consist of a mixed bag of measurements that lack consistent naming conventions. And these values often recorded with unstructured or inconsistent labels and may be missing essential details such as test descriptions, units of measurements, or reference ranges. And such inconsistencies complicate accurate interpretation and limit the ability to assess the data completeness, ultimately increasing the risk of overlooking important clinical signals. So as a result, substantial additional effort and time are required for effective data harmonization and curation.
All right, thank you, Marjolaine and Alina, for answering that question. Next question coming up here. What methods, if any, did you utilize for inputting unit values when missing?
Can you repeat it, please?
What methods, if any, did you utilize for imputing unit values when missing?
So as we said before, we created a set of rules. The data was reviewed accurately, and if we could establish the units, we would establish creating the mapping files and then coded those rules into the Python code that would transform the data based on the setup rules.
Thank you so much for that, Alina. All right, next question. How does this architecture specifically support longitudinal tracking of biomarkers and patient outcomes? And what insights has this yielded?
Can you repeat the question one more time? Thank you.
Sure. How does this architecture specifically support longitudinal tracking of biomarkers and patient outcomes? And what insights has this yielded?
So I can take the first part. So ultimately, we have it's the mixture of the real world data as well as our omics data that we're able to find and discover biomarkers across the board. And this is a ongoing process where we're looking for data, or looking through the data and finding correlations that map to different conditions, between our omics data and the real world data. And Tao, do you want to take the second part?
Sure. Yeah. As Jamie mentioned, because now we can match individual patient journey with the corresponding omics data from the serial sampling during the journey, we can make some really interesting observations. For example, for some chronic conditions, we can observe the biomarkers steadily increase over time and even before the formal diagnosis of the condition, very similar to the MASH biomarker case that I just showed. And these would be great candidates for early diagnostic biomarkers. And sometimes these biomarkers also reflect the underlying biological process and therefore can reveal novel therapeutic targets. Additionally, in individuals going through therapeutic interventions, such as medical procedures or initiation of a certain medication or even change of medications, we can observe the corresponding changes of these biomarkers. And these biomarkers could really be used for monitoring treatment response or pharmacodynamics of particular medications.
Thank you so much for answering that question, Jamie and Tao. Next question here is, what are the typical timelines and resource needs for onboarding a new omics or RWD resource into this data lakehouse? Let me repeat that.
I can take the part for multi-omics and the omics. And so for that, of course, the answer is that it always depends. More complex data sets will require more. It really depends on how many fields you have to represent in the data, as well as the number of tables. For relatively straightforward sets of data, we're looking at between a week and 2 weeks. It depends on how much pre-validation you need to do, and again, how many fields, and the overall complexity of the data. For things like genomic data, that's going to take more time. And for things like that, you will want to find third-party tools to help the process along. So around 2 weeks for typical data sets, though.
And I think I can say something about the real world data part there. And the answer is very overlapping. Like it depends on the size and the complexity of the data, and also the quality of the real world data, of course. So in general, I think you have to keep a couple of steps in mind when you determine a timeline. So you have your initial data intake, where you look at like what formats there are in the data, the volume, the quality of the data. So really see like what's in the box and how can we use it, right, for real world data. Once you have a good idea of that, you can start the mapping and define which transformations are needed. Then you need to do your ETL development and testing. The needs for that need to be set up. And importantly, of course, there's a QC step where you need to evaluate and confirm the data quality before you can continue with that final deployment of your new data source. So there's quite some steps if you want to do it right. And the first time you do this, it will require some extra efforts, but hopefully then you have that framework. And for every new source, you at least have that workflow in place, and you just need to like review the data and look at the complexity and the quality. But I think it still could, depending on the size, of course, it could still take weeks to complete that for real world data. As for resources, I'd say you need a fully functional team there with data curators, subject matter experts, data scientists, data engineers, and depending on the scale of the effort, also a project manager. Yeah.
All right, thank you for those responses, Jamie and Marjolaine. Really good questions coming in. So we have another one. For the MASH biomarker discovery, what specific analytical tools or advanced methodologies were used within the data lakehouse?
I can take this one. So in the initial discovery of the MASH biomarkers, we had to analyze more than 45,000 features from metabolomics data in 10,000 samples. And so we had to use a suite of customly developed PySpark and the Python tools to perform the analysis and of such high dimension and large scale data set. So within also the analysis had to be within a reasonable time and the computational resources. And we have since adapted these tools and modules and also added more functionality to allow us to analyze proteomics data and do multi-omics data integration as well. And additionally, I didn't show in the slide, but for the MASH biomarker, we had to investigate the genetic loci regulating these MASH biomarkers. And we performed the genome-wide association study that's by integrating with the genomics data. And this also required developing some customized pipelines, leveraging the existing AWS and the Databricks infrastructure.
Thank you for that response, Tao. Alrighty, more questions rolling in. Keep them coming, guys. What are the next planned enhancements for this platform, especially for predictive modeling, patient stratification, or novel target IDs?
Yeah, I can also take this one. So we do plan a new feature that we want to really add to the platform is the data science analytics toolkit, and especially the module for that allow us to do robust machine learning, multi-marker selection, and the model optimization. And with this tool, we can essentially, with all the organized and structured data, thanks to Rancho's expert work, we can really do at-scale analytics and develop multi-marker panels for a variety of conditions, and to allow us to more accurately predict the diagnosis and the prognosis, and for example, responder versus non-responder stratifications. The second feature that we plan to add is to continue to expand the dynamic tumor database. And we want to add more diversity in terms of cancer types and the normal tissue types and much deeper proteomics data. And particularly, we want to have the enhancement in the capability to detect non-canonical proteins and peptides that are specifically present in certain cancers, but not in normal tissues. This will be great oncology targets with less on-target and off-tumor toxicity.
All right, thank you for that, Tao. So we don't have any other questions coming in right now. We do probably have time for one more question. If anybody has any questions, any last final thoughts, comments, please feel free to drop in the Q&A box. I'll give everybody a minute or so. In the meantime, I just want to thank everyone again for joining us today. We went through, the collaboration between Rancho and Sapient, discussing how we created this data lakehouse and a little bit behind the scenes work, how we curated and harmonized the data to make these actionable insights more quick and data discovery. So I don't have any other questions coming in. If there's nothing else, then I will end the webinar. Thank you everyone for joining us. And like I mentioned, a copy of the recording will be sent to all registrants, so you can always rewatch this content later. Thank you all.
Thank you.
Thank you.
Thank you.